out, I want to go into some other detail, the structure of this genome. I want to discuss in detail the structure of genome. Should I discuss now or later? You want to learn genome now, or I directly go for application? Okay, now. So you don't have patience. Okay, let me make it a little more clear. Viral genome. It's not as difficult as you imagine to understand this viral genome. Let's suppose this is our human DNA. This is our human DNA, and here is also our human DNA, and in it this is integrated proviral DNA. Proviral DNA is having special ends by which it attaches to our DNA, These are special ends through which it attaches. These are called sticky ends. What they called? Sticky ends. They are very sticky, you know. That is why they integrate so well. They bind to our DNA. Now these sticky ends of the virus are basically, let's suppose this is 5 end of DNA and this is 3 end. Okay. Now these sticky ends of the virus, these are called long terminal repeats, LTR. LTR stand for long terminal repeat. The nucleotide sequences which are multiple repeated at the end. Now these ends of the proviral DNA, now onward I will call it just viral DNA, right? But actually it should be called proviral DNA. Now this proviral DNA, these are LTRs which are on the flanks, like my hands. And with these hands or LTRs, it stick with hold our DNA and become integrated in our DNA. So number one, it has sticky function. Number two, it has one more function. It has promoter regions here. What is it? It is having promoter region here. Promoter region is something like a lock. It is something like a lock. You know what is lock? Here the key. Now this region is locked. If you have the key, and you open it, this DNA will open and start making copies. What will happen? It will start making copies of RNA and cell will start producing virus. This is called promoter region or enhancer region, because if this lock is open, the viral formation is promoted or enhanced, or viral DNA transcription into RNA is enhanced or promoted. So in LTR there are what? The special locks which are called promoter regions, right? The multiple locks. So the multiple kind of, what are these? Multiple kind of keys can work. The keys which work here, keys which open it, once the key is applied here, and what will happen, that from this DNA, what will start making? RNA. That function is called what? Transcription. So any key, a biological molecule which opens this lock, that is called transcription factor. What is that called? Transcription factor. Here I want to tell one thing. Unfortunately, our bad luck, some transcription factor which are activated to activate our genes also work here stupidly. I mean, in our cells there are some special transcription factor, there are special keys, they are activated to activate our genes, like interleukin 2 molecule gene, right? So if this macrophage or this T helper cell, it is activated by stimulus, then that stimulus will activate the transcription factors to activate the genes of our genes. But unfortunately, same key also opens this lock, right? For example, one of the transcriptional factor is nuclear factor kappa, B, right? It is normally physiologically activated in our lymphocyte and macrophages when they want to become active. And now you imagine, if in one macrophage or T helper cell, this funny, stupid, evil is sitting there, and let's suppose this is a lymphocyte, and one antigen come and stimulate the lymphocyte, and lymphocyte want to multiply, a lymphocyte make its normal, activate its normal transcriptional factor. The stupid transcriptional factor will open the lock of viral promoter region, and our cell will multiply or not, but sure virus will multiply. And then there is another thing. Not only our transcription factor, our keys work on this, even virus can make its own keys. I will tell you later. And 1,000 times multiply its replication. I mean, it's a very deadly situation. But anyway, before I go that, this was long terminal repeats and sticky ends with promoter and enhancer regions. Then there are 3 basic genes inside it. How many? 3 primary genes. Okay. 1, 2, and 3. Right? These are 3 basic genes. Right? Into it. Now, when this gene is transcribed, and messenger RNA is made, what is made? Messenger RNA is made, and that messenger RNA is translated into protein. You know which proteins are made? The proteins which are made are P24, P7, P17. Again, this is a gene. When this gene is activated, it will make messenger RNA. That messenger RNA is a long messenger RNA, and it will make proteins. But it will make how many proteins? It will make originally one polyprotein. And that polyprotein need to be cut into 3 pieces. One piece will be P24, for example. I will show you here. From this gene, messenger RNA come out, right? From this messenger RNA, how many proteins are made? Initially, one polyprotein is made. 1, then 2nd, and then 3rd. But all of them are part of 1. So this is called one polyprotein. Then a special cutter will come. That cutter will cut at this point and this point. Now how many proteins they become? 3. One of them will be P24, other will be P7, other will be P17. This cutter is very important. We'll talk about that later. Right? This is protease because it cut. So when this gene express, right, it make a polyprotein, and when polyprotein is cut into smaller functional proteins, because this big protein is not functional. Once it is cut into smaller proteins, then we end up with P24, P7, and P17. You remember P24 was capsid, and P7 along with the P24 both are called nucleocapsid protein, and P17 is matrix protein, right? And then you must be knowing the name of this gene. What is the name of this gene? GAG. What is the name of the gene? GAG. Very good. So this is GAG gene, right? Then there is the second gene. The second gene, when this gene expresses itself, right? This was the GAG gene. Then another gene, second gene which expresses itself. Again, it also make a long messenger RNA, right? Then from that messenger RNA, multiple proteins are made. So this molecule, so we first make a very long, what is this? Polypeptide, right? Polyproteins. And then this polyprotein is cut into pieces and 3 things come out. Rest in peace. What is this? Reverse transcriptase, integrase, protease. You understand it? So now, as these genes are expressing, they start making molecule for the next generation of virus. Right? For example, there will be 10 million of these protein, 10 million of these protein, right? And material is available for millions of new virus, right? This gene is called POL. POL stands for what? Here we can remember it. It is POL is making pool of enzymes. Is that right? But again, now one thing is very important to understand here. These polyproteins need to be cut. If they are not cut, right, these molecules are not functional. They are not functional. But once you cut them into pieces, then these smaller components are really functional. For example, if this protein is there and you don't cut it, so this protein, polyprotein, will have actually, what is this? Reverse transcriptase area, integrase area, protease area. But if they are sticking together, they are not functional, right? Now, who cuts them into pieces? Yes, there is another enzyme, and this enzyme is here. This is protease, this one. Protease. This is which is going to cut into pieces. You are getting it? And make the non-functional polyproteins into functional functional peptides. Is that right? This is very important. Am I clear? Here I want to tell you one thing very important. We have another group of drug here. This is what? This enzyme? Protease. Right? We have a drug, and drug is like this. Do you think now protease can work? So this drug, what should be the name of this drug? Protease inhibitor. What is the name of this drug? Protease inhibitor. So there are protease inhibitor drugs, another group of drugs, right? You remember after integrase, we have designed the drugs which are protease inhibitors. And if we give the patient these protease inhibitors, they will go into these cells, and even if virus attach and virus fuse and virus goes through all this reverse transcription and then integration and then formation of transcription and messenger RNA and even making the what? Polyprotein. But if polyprotein cannot be cut into functional proteins, virus will be made, but its proteins will be normal or abnormal. And when virus has these abnormal proteins, we call this virus immature virus. What we call it? Immature virus. And this immature virus is non-infectious. It cannot enter into other cell. And virus cycle is blocked. So this is another way how you use. What are the name of these drugs? Yes, please. Very beautiful name. Indinavir. Yes, ritonavir. Don't call it andinavir, rotinavir. Right? This is indinavir, ritonavir. Then there is, yes, this is all about eating. Andi, roti, yes. And darunavir. Darunavir. Right? And there is another sakinavir. Yes. Sakinavir. They are not veer of anyone. Right? This is innocent protease, but with deadly action. Right? So we put indinavir or ritonavir. Don't call it andinavir, rotinavir, darunavir. It is indinavir, ritonavir, darunavir, and what is this? Sakinavir. And there are many other virs also. But this is all group of the drugs which basically inhibit the protease. And when the protease action is inhibited, you understand this is very, very bad thing, you know. Then you let the virus, but don't let it, what? Mature. It's just like that. The baby is allowed to born, but they cannot mature. Can they start action and can they, you know, I'm talking about what action? Yeah, they cannot go enhance generations. This is what happens with this virus, okay? So this was another group of drug. How many drug group we have talked about? Attachment inhibitor, yes. Say loudly. Maraviroc. It binds with CCR5. Then there was fusion inhibitor, bind with what? GP41. Drug is enfuvaltide. Then there was reverse transcriptase inhibitor, nucleotide. What is that? NRTIs. Can you tell me drugs? Zidovudine, lamivudine, didanosine. Then nucleoside reverse transcriptase inhibitor. What is this? Nevirapine. Then integrase inhibitor. What was that? Ralti graver. And then in the end you come after this duck, protease. Protease inhibitor, yes. What are the drugs? Indinavir, ritonavir, darunavir and sakinavir. Okay, but let me keep on telling the genes. So now you understand how intimately our pharmacological action is related with this understanding of genes and polyproteins, right? Now GAG has showed its action, Pol has showed. Now we are left with very important proteins. You remember glycoprotein, GP120 and formation of GP41. That is done by third gene. What is the name of this gene? ENV, right? Envelope. And this makes what proteins? It makes GP120 and GP41, but as someone was saying GP160, let me explain. Actually, when it expresses, it also makes two proteins together, right? One is GP120 and other is GP41, but together this is GP160. But it also needs protease action. But remember, this protease action is done by our cellular protease. Here we are stupid ourselves, right? But this protease was viral protease. So these two polyproteins were made into functional protein by viral protease, but unfortunately GP160 was divided into GP41 and GP120 by cellular protease. Is that right? But anyway, we only target the viral protease, because if we target the cellular protease, our own proteases are disturbed. But viral protease is different than our protease, so we can target this pharmacologically by all these indinavir, ritonavir, darunavir, sakinavir and other virs. So in this way we convert, let the virus be formed but immature. Was that right? Now let's go into, so you understand these genes now. How many? These are the basic genes which are present in all retrovirus. What are the name of these genes? GAG, Pol and ENV. Any question up to this? This was capsid protein. These two are nucleocapsid protein. This is matrix protein. This is reverse transcriptase, integrase and protease. This is GP120, right? Another thing which I wanted, and GP41. About GP120, I want to tell one more point. As I told you, this gene, which eventually makes GP120, it undergoes repeated mutations, and that makes the antigenic variants of GP120. According to the nucleotide sequence of GP120, HIV virus is also, depending upon what kind of GP120 they are expressing, HIV virus is also divided into clads. HIV1, this is divided into subgroups which are called clads, or you just call them subgroups. Again I will repeat that HIV virus, HIV1 is divided into subgroups which are called clads, C-L-A-D, or subgroups. These clads are actually differentiated from each other depending upon the nucleotide sequence in ENV gene, or the gene related with GP120. Clear? Now out of these many clads, few things are important. Three clads I would like to talk about. Number one clad is clad B. Okay, clad B. Clad B, this is a type of the virus, HIV virus, which loves to heavily infect the macrophages, and it's spread by the anal sex. So it is more commonly found in male homosexuals, and it is spreading more rapidly in America and Western Europe, right? So B clad, B for back, B for back, and B for B clad, which actually spreads. This is an HIV which loves to concentrate into macrophages, and macrophage usually transfer into male to male sexual activity, anal activity, right? Because in that the anal mucosa, rectal mucosa is very delicate and very easily tears develop, and semen with a person, if donor person is having HIV virus, right, in his semen, that will enter through those points. Then there is E clad. E clad is E for what? E clad is little different. As B clad like macrophages, E clad loves to bind with female genital tract cells. Even though all HIV virus love the female genital tract, but this one have a special love, because even small amount of this clad enter into female genital tract, it stick there and enter into the cell very efficiently. So basically, E-clad more efficiently transmits through heterosexual activity, right, between male and female. Then there is another clad. Okay, this I told you is B-clad is more in USA and Western Europe. E-clad is more commonly spreading in which countries? Specifically in Thailand. And then recently they have found there's another clad, C-clad. Oh my God, this is the most dangerous now, most rapidly spreading all over the world, especially in India or other parts of Asia and South Africa, right? And Ethiopia, C-clad. But anyway, what I wanted to mention that HIV virus is subclassified into clads depending upon the nucleotide sequence in gene related with GP120. Am I clear? Now let's come back. You may be thinking why we are thinking a lot about it, because in near future they are coming with the drugs. Some drugs will be more effective on B-clad, other drugs will be more effective on E-clad, and still others will be more effective on C-clad. So I'm preparing you for future also. But let's focus on present now. So these three are the basic genes, and these three genes are called structural genes. What are they called? Structural genes. These genes are called structural genes. Now I will give you a concept that through the same genetic material, virus can produce 6 more proteins. The question is that through the same genes, how virus can make other type of proteins, because our general concept is one gene is for one messenger RNA is for one protein. But how come? Let me explain. There's a concept of reading frames, right? Reading frames. Let me explain how is the reading frame. Let's suppose I write here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. I'm just writing something, right? Now, if I make 3 reading frames, this is one reading frame, this is second reading frame, this is the third reading frame. Just imagine this is genetic information, this is GAD, this is pol, and this is ENV. Clear? This is the basic reading frame. Reading frame means how the genetic material is read by the polymerase enzymes. Now, sometimes it happens, sometimes no, always. In HIV viral genome, they shift the reading frame. For example, the polymerase come here, it does not read into these 3 segments, not reading one unit, then second unit, then third unit. Sometimes it read like this, read like this, read like this, read like this. Now, if it shifts the reading frames, the messenger RNA will be the same as initial or different, and then proteins made will be different. So what happens with the same basic genetic information, virus can use the same genetic material to make the structural protein, and then by shifting the reading frame, it makes smaller messenger RNAs, and they make more proteins which are called regulatory proteins, and those genes are called regulatory genes. Regulatory genes. So basically, if it is, let's suppose these are the 3 primary, 1 and 2 and 3. So these are structural genes. But same genetic material, if reading frame is changed, rather than 3, it makes 6, then we call it the same genetic material is acting as regulatory genes. So basically, virus also need more proteins, but it is able to make more proteins by the same genetic information, but reading in a different way. Yes, please, you have a question.
We are told that frame shift mutation actually results in non-regulatory and non-functional proteins. Is it programmed?
It is programmed. These are very specific, not random.
Or it's a mutation?
No, it's not a mutation. Frame shift mutation in human genetic material, yes, they may end up into abnormal protein formation which are not functional, right? But this is normal function. This is not abnormal. Normally, virus plays this game that it can use the 3 basic reading frames out of this, and then it can make 6 reading frames. So how many proteins it can make? 3 group here, which are structural proteins, and 6 more group which are regulatory proteins. You are understanding? But when it is reading in a different way, then we call it a different name of gene. Am I clear? But if it's out of rather than 6, if it makes 20, then of course that reading frame shift is dangerous. Am I clear? So now we go back. Now after shifting the reading frames, the genes which are present over here, I'm just making a functional diagram. I mean, there can be more genes. The genes which are there, they can be called TAT, REV, one, two, three, 4, 5, and 6. Yeah, there are 6. TAT, REV, and then there is WIF. I will tell you exactly what these things are. And then VPR, VPU, and NEF. Okay. Right now it look a little bit burden on your mind. I hope it's also burden for the virus to remember these things. Anyway, actually, when it shifts the reading frames, it makes 6 type of different proteins. Right? Now these proteins have different function. First of all, we'll talk about TAT, T-A-T, TAT. When this, a small piece make a protein, that protein is called TAT protein. You know what is this TAT protein? This is a key. What is this? This is a key. And this will go and act here. And what will happen? Transcription will be less or more? More. So this is called transactivator protein. What is it? T for trans, AT activator. So what happens? There's a gene which makes a protein called transactivator protein, and transactivator protein go to LTR and bind with the promoter region and amplifies or increases viral transcription more than 1,000 times. What does it mean? In future, we have a drug which destroys the TAT protein. We can reduce the speed of transcription. Right now we don't have any drug. But I want my children to study this because I hope in your life drugs like that will appear. Okay? So TAT is transactivator. Clear? Then REV. REV is making a protein and it is just like a donkey. I don't know how to make a donkey, but it is just like a donkey. Right? And I don't know how to make a donkey. Donkey has horns or not? Yes. No? Okay, this is donkey you just made. This is REV protein, right? Now, this is innocent donkey. You know, this special function of this. This REV protein take the messenger RNA from the nucleus to the cytoplasm more efficiently. You remember, this is present in our DNA. So it is present in our T cell or macrophage nucleus. So here the messenger RNAs are made for these proteins.